Just of saving time, I'm not going to introduce at length George Church. You're supposed to know who he is anyway. And if you don't, you can Google him, and you don't need any keywords because he seems to occupy the half of internet anyway.
I'm thankful to everyone here and to various sources of collaboration and funding that I'll mention as we go along here. In particular, the Department of Energy and the National Science Foundation for the synthetic biology that we're doing. And it's on some of the posters, some of the graduate students and postdocs in the lab. And what we really, since we're talking about harnessing and evolving microbes, we really want to know what can and can't be selected so far, and how can we compete with 10 to the 18th liters of fluid on the surface of the planet for billions of years. And we want to be able to go as fast as possible, have as much parallelism as possible, and how radical a change can we make? So we can select for very complex structures. This is some work from John Ock in our group looking at various aspects of the 2- and 3-dimensional morphology of cells and their time series. You can obviously do that. But I think more germane to the sort of things that we want to do for biofuels and other things is looking at ways we can get a vast number of new metabolic modules. And here's an example of something that we stumbled on while we were trying to find modules that would help us deal with degradation of lignocellulose, which is recalcitrant and toxic. And what we did was figured there would be large amounts of enzymes that would deal with lignocellulose, but not necessarily with antibiotics, which was our control. And somewhat to our surprise, we found multiple isolates polyphyletically, which could in each of these, this is ribosomal and other ways of classifying the organisms, would have a resistance to not only resistance to up to 18 antibiotics, but also could subsist on them as their sole source of carbon. And this is, you can then use selection to get small genetic components, which then reveal exactly how this is achieved in these organisms when we're in the middle of it. I don't need to belabor this, but the slope of these logarithmic curves is actually going up for DNA reading and writing. It's going up about factors of 10 per year for the DNA sequencing over the last 4 years. Here's just a bit of history where we paid $6,000, or this is a purchase order from Wally's lab when I was a graduate student, $6,000 for 10 MERS. And now we pay $500 for 15 million base pairs of raw oligos. Now there's a bit of a disconnect between what you can get off of a chip and what you can get into a cell, but there already are applications for off-chip, which are pretty widespread now for short hairpin RNA, mostly in mammalian systems, and exon capture libraries, also mostly in mammalian systems. But these are actually products where you strip it off of chips. And you can think of this as kind of what Sydney was talking about, the interface between the physical world and the biological world. This is one of those interfaces where you can write DNA with spatially patterned light, and you can read the DNA in automated microscopes, such as what we used in the pollinator for sequencing. When we talk, so we have a couple of projects I'm showing here. The most minimal of these is still not intended to be a minimal genome. We're really aiming for high speed and high accuracy. Mycoplasma and mitochondria have a fairly small number of RNA and protein genes, but Mike Jewett in our group and Tony Forrester, we've collaborated with for years, is working with a system of about 151 genes of RNA and protein, which constitute all you need for DNA, RNA, and protein synthesis as far as we know, but we won't really know until it's fully functional. But this has the advantage of it allows us, it's small enough that it's easy to synthesize. It allows more radical designs than in vivo, And there are all these existing applications having to do with in vitro protein synthesis, which we all know can be improved upon significantly. So just to introduce the concept here, another reason, in addition to in vitro protein synthesis being an established market, there's a less well established, which is getting at chiral biochemistry, where you want to get a flipping of all the proteins and nucleic acids that exist in the world. And there are two routes to doing this. One is by doing pure chemistry, where you actually get pure D-amino acids, which are in an unnatural form, and then you make peptide bonds through standard peptide chemistry. And the alternative is building up a bridging translational system using chemically charged tRNAs, altered elongation factors, and peptidyl-transferase, which is a ribozyme. And this is the first one has been done, and the second one is more speculative. And Doohee Bang, postdoc in the lab, is well on his way to making a mirror image polymerase, but the other one might actually be easier in practice, although it conceptually has more uncertainties right at the moment. And so here's sort of the genetic code showing all the amino acid tRNA synthetases in the DNA and RNA polymerase, and this is the most complicated part. This is the largest small subunits of the ribosome, and this would be one of the active sites that would be changed. So we can take normal messenger RNA and put in and get out mirror image peptides. But in the meantime, we've got some very pragmatic things, which is to try to keep the high speed and accuracy of E. coli as we go into this purified in vitro systems. And Mike Jewett has very recently made some progress in terms of extending some of the pioneering work that's done by Nomura's group since 1968, extending this so that now it can be done under more physiological conditions. And so he's done it now for both the 50S and the 30S ribosomal subunits, and we feel that this is an important, possibly necessary step to get to fully in vitro replication of the including the ribosomes. Here's some of the reconstitution. We're not just making polyphenylalanine, which is relatively easy. This is a large functional protein, luciferase, and no ribosome controls or controls where we leave out the RNA or the protein from the reconstitution, showing that the subunits themselves are not contaminated with the other subunit. Now, moving to in vivo, we just heard about design and evolution. And what it is most laboratory evolution involves components of each. The more you design, the less you depend on evolution and vice versa. And here's some examples of some of our close colleagues. An interesting one is Johnson & Johnson figured out what its new anti-TB drug did, what its target was, by actually making drug-resistant forms of mycobacteria and then sequencing their genomes. I think that's a paradigm you're going to see more and more frequently. And Andy Tolan in our group has developed alcohol-resistant strains of Clostridium phytofermentans, and Xiaoxia Lin, who's here at this meeting, and Nick Repus and others have developed this tyrosine tryptophan paradigm that I'll mention in a couple of slides. And these are important pharmaceutical precursors, and so we had motivation to become familiarized with this, where we were basically looking at all the different ways that auxotrophs in two different cells could talk to one another. We did 7 different amino acid auxotrophs with one or more deletions of genes, so it couldn't revert easily. And for example, tryptophan and tyrosine synergized in a culture by syntropy, where they would exchange materials fairly well without evolution, and then we'd let them go through evolution over a period of time. And this was done in triplicate, and of course you don't expect the evolution to be entirely reproducible because you have these little jackpots in small population sizes. But as you go, we took time points along here and did sequencing, partial or complete sequencing, and each of these and 2 hours is sort of the doubling time in minimal media for the strain of E. coli, and this worked its way down from 7 hours to close to wild type. And so when we sequenced these, we found a consistent picture. Now this is one kind of evolution where you're taking spontaneous rates and you're taking them one at a time, so it has to be additive. We typically find about 3 mutations, and some of the colleagues mentioned earlier had a similar experience. Over hundreds of passages, you might get 3 mutations, and one is in the protein coding part of a pore, making it slightly less bulky and more hydrophobic, so it permitting large greasy substrates like tyrosine and tryptophan to go through this outer membrane pore, and then cis-acting mutations that upregulate this, and trans-acting repressors, knocking out the repressors that would keep this down. In addition to these that sort of were along the paradigm that we were looking for, better exchange of tryptophan and tyrosine, ultimately we'd like better biosynthesis as well. We also found that the tyrosine, eventually, very late in the process, you'd get escapees where they would come up with a new pathway for making tyrosine, and then hence would evade this cooperative paradigm where the need 2 cells in order to survive, and they would basically go back to only needing 1 cell. Now, as we're making genome engineering for metabolism, we tend not to want minimal, but more larger number of genes, not necessarily maximum, but a large number of genes. An example of this is fairly one that's at sort of at the 2.4 million liter scale now, is this production of 1,3-propionediol by DuPont, where they made 27 changes into this 5 megabase E. coli genome, and they're getting close to 90% theoretical yield. And they inserted 8 genes from Klebsiella and yeast, and did 13 knockouts and 6 regulatory mutations. And this took many years of many talented scientists. But it gives you some idea of the kind of range that we're looking at for serious metabolic engineering of 27 changes in the genome. And one of the applications is not just 1,3-propionediol, which is typically used in polymers like 3GT and carpeting, but also biofuels, which you've heard mentioned a couple of times already. And you all know all the advantages of making immiscible biofuels so that you can skim it off rather than distilling it. And you also know not to trust these nice exponential curves of increase in yield in a very short period of time, because if you start with 0 yield, you can get really infinite improvement. So if we want to improve these processes, which are now getting into the millions of liters, LS9 in that previous slide is scaling up to millions of liters, and DuPont's already there, what do we do? One of the things that drive process engineers crazy is contamination with, and so this is one of the things they worry about, are viruses, which come in all shapes and sizes, and one wonders what little do they have in common. And what they have in common is, in a certain sense, what they lack, which is they lack the protein synthetic apparatus that's complicated enough. They sort of assume that the cell is going to provide that, and they assume kind of a standard genetic code, at least for whatever host they're looking at. And so that little spinning molecule, by the way, is transfer RNA that I helped do the crystal structure of it, and that's why I like it so much. But what we're making is a new genetic code by whatever means we can, trying out a lot of different technologies. We're basically where we're starting with the easiest code change. Each of these numbers here is the number of codons in E. coli, or at least the current annotation of E. coli, and the fewest in number is 314. You can see they range up to 71,000. This is a stop codon, TAG, which we're systematically changing all 314 of these into TAAs. And this can be used not only to make multivirus resistant, but also make it harder to exchange genetically modified organism DNA with the environment, and to allow new amino acids, such as been used commercially. And I'm happy to report now, I think this is a big change from last year, is that we now have made all 314 of these in 32 strains, and actually those strains are coalescing by something I'll show on the next slide, but they're clearly viable in up to 10 per strain. This is the work of Farron Isaacs and Pete Carr and Harris Wang. Pete and Harris are at this meeting. And how we've done this, one of the main methods that we like is bringing in 90-mer oligonucleotides. This is kind of a replacement for previous methods we've used with double-strand circles or linear double-strand break repair, where you escort this with beta protein from phage lambda and somehow or another knock out mismatch repair, for example, a mutation in mut-S. And then this can kind of pretend as an Okazaki fragment in the single-strand region of the lagging strand of DNA synthesis. And this improves, this is from Quartz Lab initially, but improves recombination frequency from about 10 to the minus 4 to about 20% per cycle, but then we cycle and we get up to 100% by doing cycling. And the cycling is really very straightforward. It's just going through rounds of electroporation and cleaning up the culture, letting it recover from the electroporation, cleaning it up. Here's some parts from the prototype we have. We call this genome engineering for multiplex automated single-stranded oligos, and Harris Wang has played a big role in this. And we can use oligonucleotide pools in this process. There's a bit of a trade-off between the number of oligonucleotides that you put in and how many cycles it takes to get complete substitution or partial substitution. We've gone through a systematic survey of a lot of different parameters. I'm just going to show you a few on this one slide, where we've looked. The optimal length is about 90 mers, but with a 90 mer you can do amazing things. You can have up to 30 bases of mismatch. You can have insertion. We haven't explored insertions very thoroughly yet, but clearly up to 30 base pairs, and we've gone up to 45 kilobase pairs for deletion sizes. These are all done with 90 mers, these synthetic red 90 mers. And there's efficiencies around 20% per cycle per oligonucleotide in these oligonucleotide mixtures, but quite acceptable. So these are mutations without selection, meaning we don't have to couple this in any way with, say, drug resistance or anything like that. These will go in. Okay. So when you're using this, you can either be using it to get a specific goal, like 314 changes from TAG to TAA, or you could be trying to generate a distribution, such as shown in these two. We have 11 oligomers or 54 90mers. And the idea is to get a distribution, say you're mutating promoters or ribosome binding sites, so that you can get 1 or 2 or 3 or 4 different alleles at each position. So you're not mutagenizing the whole genome, which includes lethals and changes in enzymes that you feel are pretty optimal already, but changing the ratio of the amount of the enzymes. And here's the experiment where we're doing 32 pools of 10 to get the 314 mutations we wanted. It took about 2 days to get 250 of them, and then had to spend a lot of time working on assays for telling which is which and then combining them. So we're at the stage now where we have these 32 strains, which is collapsing down to 16 by doing pairwise recombination via conjugation. So this is the map of how we've introduced the 314 mutations in sets of 10 or so, and they collapse adjacent ones together by having, now we do use selectable markers on either side for the conjugation, and this is actually a fairly rapid procedure. But we still have a few steps before us. In principle, this can be done in 5 steps, all in parallel here. The first step down to 16, and then 8, and then 4, 3, 2, 1. Okay. So that's where we are. We haven't quite finished. Until we finish combining these into one genome by conjugation, we can't do the last thing, which is to knock out the RF1, which otherwise would be lethal. Once we knock out RF1, then we can test that, well, first of all, that RF1 is now viable, and we can determine if it's resistant to any viruses or multiple viruses, and eventually change enough of the genetic code so it's resistant to basically all viruses that are naturally occurring. So just in summary, I've shown how we can the theme here has been harnessing and evolving microbes, so we can go from raw oligonucleotides on chips, and I gave examples of how those are used, but really there's a gap between this and I think and really large libraries of useful genes or genomes, but we can introduce them to cells as raw oligos without assembly. Really, the cell does the assembly from raw oligos. In this instrument that we're prototyping and testing in semi-manual ways, this allows us to do targeted and accelerated evolution. And I mentioned about in vitro libraries allow you to get very large libraries in a very small volumes. And metagenomic prospecting, we illustrated that with antibiotic catabolic modules, but there's an amazing number of other selections that can be used and have been by companies like Diversa and so forth. So, period, full stop. Any questions? I think we've made up a little time on this, so there's some questions.
I have a question, may I?
Yeah, sure.
Could you flesh out just really quickly what is the real purpose of making a new microbe like this that has so many new different features? Is it just basically because you can, or is there a particular reason why you're making it?
If you're referring to the multivirus, so the changing genetic code has three purposes. One is to make it so that it can use new amino acids, which has value in, for example, making a higher serum stability for a recombinant for a protein drug. Also, if you make it resistant to multiple viruses, when you're using the strain in a less than perfect, very large-scale factory environment, you have one less thing to worry about, which is viral resistance. So no, it's not because it's there. I tried to give some rationale for everything we're doing. But
More questions? Well, thank you, George.
yeah, sure.